when we study cell biology. Why we are studying biochemistry? Why I started biochemistry? Because without knowing biochemistry, I will not be satisfied in cell biology. Because to know that, when I will tell hemicellulose. Hemicellulose is made up of arabans and xylans. At that time people will know what is this? Arabans and xylans. But now you people will not be surprised. Arabans means polymer of arabinose and xylans means polymer of xylose. That's what I am describing here. Now derivatives of hexoses. Because when we say chitin is a carbohydrate present in the exoskeleton of insects. And N-acetyl glucosamine, it is present in the bacterial cell wall. So all these things, if we know this, only then we can understand their inhibitor. There we have to put extra effort. If we are knowing these things, we will understand all those things very easily. And many things, drug development thing will also be known to us here. How molecule changes and what happens we will understand today. A little bit, not much more, later on. When a hydroxyl group in the parent compound is replaced with another substituent, when a carbon atom is oxidized to a carboxyl. Now all the time I will not without making the structure, you will not distinct. This is what? Alpha D glucose, isn't it? Alpha D glucose. And if we change it because it is abundant in amount, then what is beta D glucose? You start numbering first, second, third, fourth, fifth. This is sixth carbon. The first carbon is hemiacetal. First carbon is hemiacetal. If this OH is replaced by amine or amino group NH2. Now it is what? Glucose plus amine. Glucosamine. What is it? It is glucosamine. It is on the slide also, but without making whole picture. It doesn't. So that is glucosamine. The same thing. If it is replaced in galactose, then it is galactosamine. When it is replaced in mannose, then manosamine. Understand? Yeah. Prasad. So glucosamine, galactosamine, manosamine. Replaced with another substituent or a carbon atom is oxidized to a carboxyl. When a hydroxyl group, one more thing we have to understand. When a hydroxyl group in the parent compound is replaced with another substituent, or a carbon atom is oxidized to a carboxyl group, that is also, I will describe it later. So this is your OH group replaced by amino. On that basis it is When hydroxyl at second carbon of glucose, galactose and mannose compound is replaced with an amino group, glucose, galactose and mannose.
So why for mannose and when it is different means the amino group is inside.
Because from mannose, what is the different? At second and third OH will be opposite direction. No? Therefore, between the glucose, third, when you say galactose, so both will be OH and H. No? And then it is galactose. So in the case of galactose, NH will attach here. No? Therefore, it is there. Now you what? Now, N-acetyl glucosamine. This is your NH2 and this is your acetyl group. Then we say CH3CO. What is this? Acetyl group. Particularly in the case of epigenetic regulation, there is acetylation. So acetylation means this is your what? Acetic acid. When this acetic acid will react here, okay, it binds with your if you break NH2 in this form, OH and H, remove one molecule of water, then it will attach with your amino group. Acetic acid will attach with your amino group. Here OH, H of NH2 and OH of carboxylic group is removed, one molecule of water forms, then this molecule is what? N-acetyl glucosamine. That is your N-acetyl glucosamine. Okay? Where it is present? Bacterial cell wall. N-acetyl glucosamine is present in your bacterial cell wall. Now, N-acetyl muramic acid. Lactic acid is either linked to the oxygen at the third carbon. You can see here. Third carbon. R is what? Lactic acid. And acetyl glucosamine, it is also present in bacteria. That is different name. Acetyl muramic acid. Here this and acetyl glucosamine, and when a lactic acid is attached at the third position, then it is called N-acetyl muramic acid due to presence of that lactic acid at the third position. Now, L-fucose. Substitution of a hydrogen for the hydroxyl group at 6th carbon of L-galactose. Galactose is this, isn't it? You remove from here. There is what? CH2, OH and H. If you remove what? Hydroxyl group. If you remove this CH2, OH, CH2, OH. If you remove OH, then it is your L-fucose. Hydroxyl group is not there. And the same thing